computer. And there we go. Okay, in that case, I will share my screen then. And this time we want to make sure I optimize for video. Okay, can everybody see that okay? Has that worked?
Yep.
Yeah.
Thanks.
Okay, so this is part 2 of talks about species distribution modeling. And this will be a little bit different because some of the stuff I'm going through takes a very long time to run. And so what I've actually done is recorded the various bits I was going to show you and actually edited them down to edit out the gaps and speed up some of the longer bits. So I'm going to show you, we'll go through the general ideas, and then I'll show you 4 bits of basically screen capture movies. One of them took me more than an hour to actually run, so it edited down to about 16 minutes. So you can see that some of these things take a good deal of time. But anyway, last time we looked at the general principles, which is taking the known distribution of the species, the known points of presence, taking various environmental layers, essentially maps of some environmental feature, building a model training a model which attempts to say what's special about those points of presence, what's distinctive about them, then apply that model back to the whole layers and end up with a potential distribution. And so the steps are getting the known species occurrence, assemble a suitable set of environmental layers, train the model, and then evaluate it. We saw last week one possibility is you split the available presences into 2 sets at least, and use the one, withhold some of them from training the model and use those to actually evaluate the model, see if it's any good. Now, last time, what I actually showed you was simply running Maxent straightforwardly, loading data, which we dumped out of the Hoverfly Recording Scheme, accepting all of Maxent's defaults and letting it do its thing. And then we saw how to look at the results. What I want to do this time is actually go through in a little bit more depth. So we need to be able to run Maxent sort of programmatically, so we can then manipulate it a bit more and look a bit more of it. So this first screen capture just looks at running Maxent programmatically from R. So I'll start it.
Okay, we will look at programmatically fitting a Maxent model. So you've seen the first part of this before. We're going to load the data table and lubridate packages to load the data, and we're loading this package called Dismo. Stands for distribution modeling. Let's go ahead and do those things. Notice that the Dismo library load packages called raster and SPOA, which is various spatial procedures. We'll see those again in a minute. So we're going to work on Chrysotoctin arcuatum again. We're going to look at the last 10 years of data, and I'm going to select 5,000 background points. So we load the observations, extract years from them, and extract the 1km grid references as we've seen before, and we'll load the coordinates of 1km squares and we'll join them. So now in our observations we've got XY coordinates. So we can get the presences of our species. So we're filtering it to the particular species. in the year range we've selected, we're asking just for the X and Y column. We're getting unique values, and we're making sure we throw out any that have got missing data. So that's 517 presences, and they should just be the XY coordinates. We're going to divide those presences into two, and we're going to use this procedure called kfold from the Dismo package. We're passing it the presences, and we're telling it how many folds we want, which is just 2. And what that'll do is return this variable called folds, which is the same length as our presences and is just categorized into 1s and 2s. So if we just have a quick check that table folds, we can see 258, 259, there are an odd number of observations. And we need to load our environmental files, and we've seen this before. Now the other thing we need is a random sample of background squares, and there is this procedure called sample random in the raster package, which, given a raster layer or a stack of layers as we've got here, we can tell it how many we want to sample. We can tell it we want to ignore missing values, which in our case are going to be squares in the C. I'm telling it I want the XY coordinates returned, and I want it returned in the form of a spatial pixels data frame. So we can go ahead and do that. Right, well, we'll check what we've done so far. So I'm just loading a GB outline. We'll plot it. This parameter asp controls the aspect ratio, so I'm saying that the X and Y scale should be the same, otherwise Britain would look in shape. And we'll plot the background points in grey, and you can see this random scatter throughout Britain, not including the Isle of Man, because some of the environmental layers are missing for the Isle of Man, as we've seen before. And here are the first lot of presence points. If we say folds equals 1, that's going to be our training data. And then if we say folds are greater than 1, that's going to be our testing data. So you can see now we've got the random spread of background points, we've got the red points we're going to train it with, and the blue points we're going to use to test it. So we're ready to fit a Maxent model. And we do that with the Maxent procedure from Dismo. We need to pass it our environmental layers, our presence points and our absence points, and this procedure wants them as data frames. Presences were actually data tables because we were using the data tables package, and the background were this spatial pixels data frame. So in both cases we need to convert them to data frames, the format procedure wants, by using this as data frame procedure. We need to tell it which of the environmental factors is categorical, and if you remember, my standard thing is to name them starting with cat underscore. So this says find layers that are named starting cat underscore, and those will be our categorical layers. And I'm passing it a path, which is where it's going to store its results files. So let's let it do that. Okay, now we're going to evaluate our model, and this time we'll pass it, instead of the presences for fold greater than 1, remember this is our half of the available data we're going to use for testing. And again we're going to pass it the background, we need to pass it the model we've fitted, and we need to pass it our environmental layers. Okay, and now we've done that, we can recover the from this evaluation object we can recover the AUC, and we can also calculate various thresholds by passing this evaluation object and what threshold we want. So we're just going to print some of those results, and you can see we got a pretty good test AUC and a threshold of 0.02. And now we will predict the potential distribution by applying the model to the whole of the environmental layers. Okay, so we will have a look at the results by plotting this potential distribution, I will just put the outline of GB on that to make it look a bit solider. And then we could apply a threshold to, again, remember this is the same threshold we saw a minute ago. We'll apply that by asking for the places where our predicted distribution is greater than equal to that threshold, which will give us sort of a predicted presence-absence distribution. And again, put an outline around it. Okay, so that's how we go about programmatically fitting a Maxent model.
Right, well, we can just have a quick look at the results we got. So remember that I gave it a path to Maxent results. So that should have put the results in that path, which it didn't. Oh dear. Because I was running a screen movie, of course, it didn't. Sorry about that. Oh well. Daft, that's what we get. Oh well, doesn't matter. But I'd point out that we made a couple of assumptions really. We're assuming when we're doing this that the presence points we provided are a representative sample of the requirements of the species. And we're contrasting that with a random sample of background, and and that contrast between the places where the species occurs and this random sample of background is trying to characterize the places which can support the species. But the trouble is, we know that the hoverflies collated by the recording scheme are pretty geographically biased. So this is that last 10 years of data, and it's the density of records contoured out. And you can see they tend to come from the lowlands of England particularly. And you can really think that instead of this a set of presences providing a representative sample of the requirements of the species, it's actually a sort of joint distribution which will sort of represent hotspots, because that's where people tend to go to find hoverflies, nature reserves and so on. And secondly, the road system, because that's what people use to access places. So it's not really a very representative sample of the background. If we just go back to that a second, the problem arises if we now take our random sample of background, we're going to sample this area, which is really poorly sampled for hoverflies, as much as we're going to sample this area, which is really well sampled for hoverflies. And that's going to accentuate the differences between this part of the country and this part of the country because of uneven sampling, not because of differences in the species. In other words, we're going to bias things. And what we're actually likely to do is over-egg the distribution model. It's likely to produce a higher difference than is really true. So how can we try and get around that? And there are really three basic ways that have been suggested. The first is to try and fix the samples by some sort of subsampling to try and achieve a more even coverage. This is known as thinning, and you can try and thin geographically by, for example, selecting a subset of points that are as far as possible from each other. Or you can do it environmentally by thinning the records by choosing ones that are most environmentally different to try and represent the sort of range over which the species occurs. Problem with this is you're throwing away a lot of your data. The only data you've got about the species really are these presence records. And especially if you don't have many presence records in the first place, once you've done this, you don't have much left really. So I've never found this a terribly good way. Second thing we can do is try and bias the sampling of the background to match the bias of the presence records. And one way of doing that is to take the all the places that the Hoverfly Recording Scheme has visited and actually take your sample from that subset of visited squares rather than from the whole map. And therefore that should give you a background sample that's similarly biased towards the southeast and away from the northwest, the same as the species record is. The third way you could possibly do it is to provide a so-called bias layer as one of the inputs to the analysis and attempt that correction computationally. And potentially that could be a bias layer. You'd actually use some sort of another raster layer like that to say, this is how the data's biased. Personally, I've not found that very successful. Maxent does have an experimental feature that lets you do that, but I've not had a huge amount of success. And the published research really favours this middle method as the one that produces the best results. Another potential source of bias is including samples from large parts of the countryside where the species doesn't occur, because again, that'll tend to overcook the contrast between the points of presence and the background. So really, you want to take the background sample from the area over which the species might be expected to be distributed, and not sample background points from, areas that are completely unsuitable for the species. That's known as a calibration area. And one possible way of getting that is to stick a polygon around the areas that the species is known to occur from, possibly stick a buffer around it, and then do your sampling from that area. So let's see how we can actually go about utilising those couple of ideas to try and produce possibly a bit better model.
This time we're going to fit a model using Dismo again, but we're going to try and mitigate some of the biases. We're going to use this package called FlexSDM, which has a procedure which we're going to use to get a calibration area. So let's go ahead. Same as last time, price toxin marcuatum, last 10 years of data and 5,000 background squares. Again, load the observations exactly the same as we've seen before. So what we're going to do is we're going to get a data table, I'm calling it all square, which are the 1 kilometre squares from which we've received data during our period. So that's all the squares for which we've got data. You can see there's 34,000 odd of them. And again, these are just XY coordinates. And we'll get the presence data exactly as we did before and load the environmental layers. Now, we're going to use this procedure called Calib area from the FlexSDM package to actually select an area that encompasses all our presences and puts a buffer around them. To do this, it needs a raster layer to actually get the geography, to get the coordinates in the right sort of geographical space to actually fit these in. So I'm going to load a raster layer which actually is just regions of GB. You'll see what it looks like in a minute. And I'm going to use a method which is actually a convex hull around all the presences with a buffer around them. And I'm setting the buffer width to 50. Now remember, we're working in kilometres of the OSGB national grid, so that's 50 kilometres. And it's going to get the coordinate system we're using from this reg raster layer. So let's go ahead and do that. We'll load our reg raster and get a calibration area. Now I want to select the background points by taking a random sample from this all squares, so all the squares from which we've received hoverflies, but I only want it to be the ones that are within this calibration area. So the first thing I need to do is turn this calibration area into a series of XY coordinates. And there is there's a function called CRDS in the Terra package, which is a package that FlexSDM likes to use. So we got access to it because we loaded FlexSDM, and so I can call that, and I can turn our calibration area into a data frame rather than a list by setting df true and list false. So let's do that. So hopefully that's now just a list of coordinates. And what I'm going to do is use a function called pointInPolygon from the SP package. Remember, that's loaded by our Dismo package, which finds out which of these all square points are within this polygon. So I need to pass it the x and y coordinates of the points and the x and y coordinates of the polygon. What that'll do is it will return a list which has the value 0, 1 or 2. 0 for points which are outside the polygon, 1 for points that are completely within the polygon, and 2 for points that lie on the boundaries of the polygon. So if I'm saying I only want the ones with a value 1, then those are the ones that are entirely within our polygon. So that's selecting from this all squares data table those points that are completely within that polygon. So our background is going to be firstly the squares that are within that polygon. So from the 34,700 we've now got 20,500 which are within that polygon, and we're going to take a random sample of those. So we can use this procedure called sample. We tell it the number of rows in background, and we tell it what number of those we want to sample, and that's set to 5,000. So that's asking to sample 5,000 rows randomly from the 20,500 that are in that list. So we should now have a list of 5,000 random coordinates from that all squares thing that are within that calibration area polygon. As we saw before, we'll split our presences into two folds, and we'll visualize them again. So we're plotting this reg layer, which as you can see is regions of Great Britain. We'll plot our calibration area, so that's our calibration area. If I put the presence points on it, again red points and blue points, you can see here this calibration area is a convex polygon encompassing them all with a buffer around it. And if we then plot our background points that we selected as grey points, you can see they all lie within this polygon, and they're also, instead of being widely scattered throughout the area, sort of evenly scattered, they are tending to be clumped where our sampling is most clumped, because they were a sample of all the squares from which we've had hoverfly records. So we're hoping they're biased in a similar way to which the presence points are biased, and that's the whole point. So we'll go ahead and fix our Maxent model exactly as we saw before. Again, as you can see, we had to use as data frame. We had to give it the factors with names starting cat. We'll evaluate it exactly as we did before. We'll print the same set of results, and you can see the test AUC has come down a bit. Hopefully that's because we've removed some of the bias, so we're not over-egging the difference between the background sample and our sample of presences. And we'll predict the whole distribution as we did before. and we'll visualize the results as we did before. This time I'm loading an outline which doesn't include the Isle of Man, because if you remember, some of our environmental layers don't cover the Isle of Man. So we'll plot the resulting distribution, put an outline on it, and we'll plot the distribution thresholded as before and put an outline on it.
Okay, so that is a means of trying to mitigate some of the bias. The other thing we did so far today and when we ran Maxent last week was we simply accepted Maxent's default parameters. Maxent actually has a series of parameters which can be changed. The factors are how the environmental variables are treated and turned into things that are actually going to be calculated by Maxent. You can have a linear relationship between the environment and what goes into Maxent modeling. You can have a quadratic relationship, put in squares and so on. You can have a product relationship, so you can multiply together pairs of environmental variables to look at interactions. And there are others as well, as we'll see. Second parameter is the so-called regularization. This is a value that applies a penalty to more complex models. It's a way of fighting against overfitting. The higher the regularization value, the more complex models are penalized. And finally, we can tell it how many iterations to undertake. This is the number of iterations the algorithm goes through in trying to converge. And the higher the value, the longer the fitting process will take, but the more finely it'll converge. And again, research has shown that these variables do make quite a difference to the quality of the model. But if you actually do a literature search, you find that the great majority of authors simply treat Maxent as a black box and accept the defaults. So a lot of people don't really bother with this. And other machine learning models that we might go in for, like random forest and boosted regression trees, have their own sets of parameters that, again, you need to optimize, although regularization and iteration tends to come up fairly commonly. And the literature shows people tend to just accept the defaults with those as well. So if you're going to try tuning the model, the simplest method is really just trial and error. It's called grid search. So if we choose some possible values of each of those three parameters, if we chose, say, six possible values of each, that'd give us 216 combinations. And grid search just simply fits 216 models, evaluates it against the withheld data, and chooses the set of parameters which gave the highest evaluation score from this testing. And as you can imagine, that takes quite a while because you're fitting lots of models. But it is very parallelizable, by which I mean that if you've got multiple CPUs in your computer, it can run a separate model on each of those CPUs. And so my particular computer has 12 CPU cores in it. So potentially it could run 12 models at the same time, hence reducing the overall time. Oops. There are other methods, genetic algorithms. They use sort of natural selection-like processes, including mutation and selection, to try and find the best model in fewer steps. But grid search is pretty easy to understand. The other question that comes up is, finding the best set of environmental variables to use for a model. Now, ideally, what we'd really like to do is have a good knowledge of the ecology of the species and therefore choose appropriate environmental variables that actually matter to that species. Trouble is, in many cases, we simply don't know enough about the species. I mean, roughly half the British hoverflies have reasonable knowledge of their larval horticology, so that we could, with some guesswork, come up with, a short list of environmental variables. But the other half, we don't really know that much about, so it'd be a bit difficult. So a very common approach, and the approach you've seen so far, is get all the environmental layers you can lay your hands on, throw them all at it and see what sticks. And one potential problem with this is that environmentals are quite highly correlated. So altitude and mean temperature, soil type and land cover. This can cause quite a problem for some methods, particularly regression-based methods, but researchers suggested it doesn't really seem to be a problem for machine learning models. They simply pick and choose amongst the variables you've given it, and they don't seem to be bothered by cross-correlation of this sort. But even so, it would be nice to achieve the most parsimonious model possible. Occam's razor, so good old William of Ockham, and all things. Now, one way of doing that is a technique known as jackknifing. You fit an initial model with all the variables in, and then you try a series of models where you drop one or a few of the variables at a time. You go through this process of dropping each of the variables. Again, you evaluate the resulting models against data that was withheld from the training, and you find the one where removing a variable has made the least difference. And then the variable you remove to get that model you drop, and you go round again, and you keep doing that until you reach some stopping condition, like the model has degenerated unacceptably if you the model will degenerate unacceptably if you remove any more. Again, it's an effective method, but it takes a lot of computing time, and again, it's eminently parallelizable. You can run these various models concurrently on different CPUs. So let's have a go at tuning a Maxent model, and this is the one I mentioned earlier. Took me, I think, about just over an hour to run. I've edited it down to 16 minutes, and some of the bits in it are running at a round about 100 times real time. So enjoy.
Okay, we're going to look at tuning a model. I'm going to use a Maxent model using this package called SDM Tune. So I'll talk you through this, but because it's actually a process that's going to take a long time, I'm recording this as a screen movie, and I'll edit it to take out the long waits in it. So we're loading data table to handle a large data frame, the SDM Tune package, and we're loading a library called Terra. Now this is a sort of updated version of raster that's preferred by some recent packages. So we're setting our usual directory, we're clearing variables out, and we're setting up some parameters. So we're going to use data for a 5-year period from 2015 to 2019 to model. We'll use the center year of that period, 2017, for our environmental layers. I'm going to look at Leucozona glossia, and I'm going to do a sample of 5,000 background points. So let's get this going. Make sure all this happens okay. Okay, I've set up two functions. One reads the environmental layers, so it develops a list of files which I first populate with the altitude and the soil, which of course are fixed, and then the ITE land class and the bioclimatic variables, which differ depending on the year. So we feed it the year we're interested in and it reads the layers for those particular years. We load those files into a raster stack, and there's a peculiarity of this SDM tune package that some of the procedures don't seem to like numeric characters in their layer names, specifically negative sign and 0 to 9. So once I've actually loaded the stack, I'm modifying some of the layer names to turn minus signs into underscores and to get rid of the characters 0 to 9, and then we return that stack. Just make sure we've loaded that. There we go. And the second function here, which simply shows us the results of a model. So it shows us the training AUC, the test AUC, predicts the model across the entire layers and shows us the plot, and it actually returns the predicted raster layer. So we'll read the observations. So there's 1.5 million of them. We'll extract the year from the date, clean up the grid reference and extract 1 kilometre squares. We'll load 1 kilometre square coordinates and join those together. So we now should have observations which have got the XY coordinates in kilometres of our 1 kilometre squares. Right. Load our environmental layers, and we can check the names of the layers. We should see that for example, this one, the 85 has gone from the name, and ones like this one, supralittoral, has now got an underscore, so it doesn't upset our procedures. Load the presences for that species. So these are the observations for that species within our year range. I'm getting the XY coordinates, I'm getting a unique set of them, and I'm removing anywhere they're NA, so where there's missing data. And the same for all the 1 kilometre squares from which we received records, which is exactly the same query, except I've removed the filter for the species name. So we had some 20,000 odd 1 kilometre squares with records and 937 presences. Now the first thing I've got to do is set up a data structure which has got so-called species with data, or an SWD data frame, and I'm passing it the environmental layers, the presences, the coordinates, the present points, and the coordinates of my background points, and I'm going to take a sample of background, which remember was 5,000, out of those 20,000 as my background. I also need to tell it which of the layers are categorical, and remember I prefix my categorical layers, which in this case the only one is the soil, dominant soil type per 1 kilometre square, with the term cat underscore. So this says find names starting with cat underscore, and those are the categorical ones. So if we run that, it's going to get the information for the presence and the background layers, and it's going to return a data frame with that information in. Okay, so 49 of the presence locations were NA for some environmental variables, and 347 of the background. Now these are going to be Isle of Wight records, because remember the ITE land cover data isn't available for the Isle of Man, sorry, not Isle of Wight. So we're going to be left with around about just under 900 presences and around about 4,600 background points, which is fine. I'm going to split this into 3 random partitions, or the present, I'm only present, so the presence is into 3 random partitions, which I'll use for training, testing and evaluation of the models, and for validation, which we'll come back to. So let's do that. And then I'm going to fit a Maxent model using the first of these 3 partitions of the data, so the training partition, which is going to be roughly 200 presence observations. And we'll take a look at the results, and you can see the training AUC was 0.87, which is quite high, and the test AUC was just under 0.8, so 0.79. And there's our predicted map, which looks reasonably good. So the first thing we're going to do is we're going to go and train the model by tuning its parameters to give us the best possible testing AUC. And the 3 parameters for a Maxent model are the regularization, the factors, the way the model actually combines our environmental variables, and the number of iterations the model goes through. Now the regularization in this case I'm not going to vary, because my past experience of doing this is if I vary the regularization, whatever the maximum value I give that range, and it can be a silly huge value, it'll always come back with the maximum possible value of the regularization. So it seems a little pointless. The way it forms factors, Maxent can use a whole array of different ways, and these are designated by letters. So L means linear, which is just linear combinations of the variables. Q is quartile, which is squared combinations of the variables. H is hinge, which is sort of broken stick regression. So it's linear, where it allows the line to change slope at some point. P is product, which means pairs of variables multiplied together. And finally we have T, which is threshold, which it applies a threshold. So you might imagine something like mean temperature. There might be a minimum and a maximum temperature at which the species is active. So actually applying a threshold before you then apply the value makes sense. So we're trying combinations of just linear and quadratic, linear and hinge, linear, quadratic and product, linear, quadratic, product and hinge, or all the possibilities added together. And then the number of iterations I'm allowing between 500 and 2000 in steps of 250. And what we're going to do is brute force this and use a grid search, which will try each of these possibilities, and it will find the maximum possible AUC. And for the evaluation of each of these models, I'm going to use this third set, I call the validation set, so that we can then test the resulting model independently of what was used, in actually generating these things. I'm also setting save models to false, because if there's going to be quite a lot of models. If we run that param, we can see we've got 6 different factors, 7 different iterations, so we're going to run 42 different models. If I keep them all, that'll be a pretty enormous memory structure, so I'm going to avoid doing that, and I'll actually find the one that was best, and I will just rerun that individual model. So this is going to take a long time, so here we go. Okay, well, as you can see, that took a quarter of an hour to run, and we can see from the plot up here, the orange points are the training AUC and the blue points are the test AUC, that the 1, 2, 3, 4th step each time was remarkably poorer than the others, which is the 1, 2, 3, 4, the factors with linear, quadratic and product types. But there isn't an obvious effect of the number of iterations. But anyway, we can look at which model performed the best by looking in the results for the maximum AUC, validation AUC, and it was model number 33. So if we now look at the factors, it's linear and hinge, and the number of iterations, it's 1750 iterations. So let's fit a model using those parameters, and let's have a look at the results. Now if we compare that with what we had before, we had a test AUC of 0.79, and we've improved on that a bit. We've gone up to 0.805, so we've done a bit better there. We're just waiting for the map, and I don't think that's very noticeably different. We don't seem to have the previous map. So the second thing we're going to do is eliminate environmental variables which don't contribute all that much. So what this procedure does is, again, the metric I use is the AUC. Again, we use the validation set for testing, so we've got an independent way to test it. I've set a threshold that if the model doesn't, if an environmental variable doesn't contribute more than 2.5%, then it's a candidate to eliminate, and we're going to use 10 permutations and jackknifing to actually test these things. This again is going to take a long time, so we'll get that running. Well, I'm afraid that that doesn't show a progress bar and give a time count, but I can tell you that took just over 23 minutes, and you can see the variables it's reduced here. So we'll have a look at what the results are like, and you can see that the test AUC has gone up a tiny bit again. So if we go back to what it was initially, at the last step it was 8049, initially it was 7914, so we're about up to about 81%, and I think you'd agree the map probably hasn't changed a great deal. We can actually have a look at the variables that are still in there. So we're left with 14 variables out of our initial, can't remember, about 42 I think. Those are the ones that are still in. So we'll save that predicted model as an ASCII file, and we'll save the fitted model so we can use it later. And that's about the end of that procedure.
Okay, so we've seen there how we can actually tune a Maxent model, and by actually tuning the parameters and eliminating some of the environmental variables, we did actually manage to make the improve the test AUC, both of those steps. So the final thing I'd like to look at is the idea of ensemble modelling. And there's a couple of quotes here, one from Science Direct and the other from Wikipedia. So an ensemble model is a process where multiple diverse models are created to predict an outcome by using many different modelling algorithms and/or different training data. The ensemble model then aggregates the predictions of each of the base model, resulting in one final prediction. And Wikipedia says in statistics and machine learning, ensemble methods use multiple learning algorithms to obtain better predictive performance than could be obtained from any of the constituent learning algorithms alone. So the idea is we can fit several different models. We can use different modeling techniques. We can use different samples of the presence data and the background data to produce a whole series of models. And then in some way we combine them, hoping that the result will be a better, more stable result than any of the single models. So we can have a look at how that works. And this is the last of these videos.
So we're going to look at fitting an ensemble model. That's running several different modelling methods and then combining them together to get hopefully a better answer. So the package we're going to use is Flex SDM, and it uses the Terra package to deal with the raster layers. And I've set up a directory with this information in, and as normal I clear out the environment. So let's just run those. And the parameters, so I'm going to look at Leucozona glaucia. I'm going to use a 5-year period as before, 2015 to 2019. I'll use 5,000 sample of background points, and I'll partition the data into two, so a training set and a testing set. And we'll use a 40 kilometre square to form a calibration area as we've seen before. So if we set those up. Now I've defined here a function to actually allow us to visualize the model, and since we're going to be fitting a whole series of models, I want to do this several times, so it's sensible to define it as a function. So we're passing the fitted model to the function. First thing we're going to do is predict the potential distribution using this model across the whole of the layers, and we'll also predict a couple of thresholded pictures as well. Once we've got this predicted layer, so it'll predict the logistic probability and the thresholded, these two thresholded things. So I'm using here the first of those, which is going to be the logistic probability, and I'm extracting the predictions for the presence points and the second partition, our test partition. So I'm extracting the XY values, and I'm getting the values, the predicted values at those particular points. Same with the background. So again, we're using the background data for the second partition, getting the XY coordinates and finding out the prediction at those points. We'll then run an evaluation of the model using those as presences and absences, and finally print the AUC from that evaluation and actually plot the raster map to show what it looks like, and the function returns that fitted raster map. So hopefully that should be all work fine. So now we can start doing something. So we read in our data, which is species, year, XY coordinates. First thing we want is a list of all the 1 kilometre squares from which observations were received, and we're going to take a random sample of those as our background. So as you can see, there are 20 odd thousand of those, and we want the presences of our species during the period we've defined. So and there were 937 presences, and we need to load the layer files. Now I've set up a directory which simply has those layers that were selected when we did the layer reduction. So it's just 14 of those layers. So we get a list of the files, and we can't actually use categorical variables in all these models. Maxent can certainly use them, and I think one or two of the others may be able to, but several of the models we're going to fit will only work on continuous variables. So we need to remove that categorical layer. So I found it, and I've put a minus in front of it to actually remove it from this list of files. So if we look at that, we should see it's only 13 now, and the soil one starting with cat underscore is gone from there. So we can lower those as a raster stack. For some of these procedures, we actually need to put a unique identifier on each of the rows. So I'm adding an ID column to the presences, which is simply going to be a number running from 1 to however many presences there are. So if we look at the presences, you can see there are just X, Y's. If we run this operation, they're now ID and X, Y, and each of the rows is simply numbered 1 to however many there are, 937. So we'll, as we saw before, we'll do a calibration area. So we load the region file and get a calibration area using a buffer, and we'll just have a look at what that's produced. There's our region file, and there's our calibration area, which for Lucozone and Glaucia is pretty much the entire area of Great Britain. So what we're going to do now is to filter our presences to get a subset which are as widely dispersed as possible across the environmental layers. And that found 371 presences, and we can have a look where they are. So hopefully these are now more widely dispersed, they're thinned out. And we need to extract the layer data for them and add an adder column indicating presence absence. I've called it PR_AB, and it's going to be 1 for presences, which these are, and 0 for the background. So if we now look again at that variable, we've got all these environmental variables, and we've got a PRAB column which will be 1 in every case here. Okay, we're going to do the same for the background, so we'll take a random sample of all the squares from which records are received. We're going to do number of background points for each of the partitions. It's a random sample, and we'll restrict it to our calibration area. And we need to load the environmental values for those points. And again, we lost a few of them because of the Isle of Man, so there are just under 10,000 of them there. So now both of these things, the presences and the background, we want to partition them into 2 random partitions, which are going to be our training and our testing sets. So we can do that. Now for most of these modelling methods, we actually want all this data, both the presences and the background, in a single data structure. So I'm simply going to add those 2 data tables together, the background and the presence data, Unfortunately, the order of the columns in those two is slightly different. You'll see we've got IDXY on that one, and in this one we've got no ID, sorry, wrong one, this one. We've got no ID, and we have the presence absence before the environmental variables. So we have to rearrange the columns slightly, which is what the slot does here. So we should be able to have a single data frame which includes all of the variables now, and you can see there it's 10,313 observations, and we can just test that's worked okay by doing table SWE PA AB and SWE dot part, and there we can see we've got for the two partitions and for presence and absence, we've got roughly 5,000 background points and 180 odd presence points for each of those sections of the data. So now we can fit the models, and for some reason Maxent seems to require you provide the presence data and the background data separately. According to the documentation, you ought to be able to provide data with this PRAB column with noughts and ones in it and leave the background null. But whilst that actually works, the model it produces is just empty. It doesn't have any values, so for some reason it doesn't work like that, and you seem to have to provide the presence and background data separately. I've set things up so I can do that. Okay, we'll have a look at the result using our show model function there. So there's our Maxent model, and again the two thresholded pictures. So now we're going to fit a boosted regression tree, a random forest, a support vector machine, and a neural network. And all of these you can simply provide a single data frame, this SWE, with a response column that has 0 and 1 in it, and also the partition column, and that actually works fine. So I don't know why that doesn't work for Maxent, but anyway. And again, look at our results and the random forest, support vector machine. For some reason the values on this are all very low, so on this plot they're hard to see, but you can see when they're thresholded the picture is not much different from what we've seen before. So finally a neural network, and again look at the results. Okay, so we've now fitted 5 different models: Maxent, random forest, a neural network, a support vector machine, and a boosted regression tree. And the whole point of this exercise really is to fit an ensemble model, which is a combination of these, and we can combine these results in a variety of ways. The one I'm going to use here is a weighted mean, so it will take the mean predicted result for each of these things, and it will weight them using the AUC. So the better the model, the more it contributes to the final result. The other possibility is you can actually do a thresholding model like this. So it's predicting presence or absence using a particular threshold for each square, and then it essentially votes on those results. So it will predict a square if the majority of the models say it should occur in that square using this particular threshold. So let's see how that works and have a look at the results. And we can see that the picture we get is a pretty familiar one. The AUC is pretty high. It's really it seems to have done a remarkably good job, really. And we can see that the picture of the distribution of the species looks quite compelling. So that's an ensemble model.
Okay, so uh, that covers the things I was wanting to cover. So we looked at programmatically fitting the model. We looked at the various biases these models might be subjected to and how we might mitigate them using a calibration area and a sample from the squares from which hoverflies have been received rather than just across the whole background. We looked at tuning the model, and then we looked at fitting a whole series of different models, and I could have perhaps fitted 10 of each of those models with different samples of the presence points and different samples of the background, and then producing an ensemble of them. And I think as we went through each of those steps, really improved the test AUCs we were getting out of it. So the general fit of the model. I didn't mention on the way through, one of the reasons I switched to Lucasona Glaucia for the tuning is because, as you saw, the calibration area is the whole of Britain. So I could ignore the calibration area step for that one, which just made it a little easier. But anyway, so hopefully that that covers what I was intending to say this time. And the final of these 3 talks in 2 weeks' time, which hopefully might be a little bit shorter, I just want to consider one of the possible uses of this sort of model, which is forecasting. So we're in a time where the climate's changing. One of the questions that keeps getting asked is, how's that going to affect our species? And fitting a species distribution model and then applying it to predictions of the changing climate is one way we can investigate that. So we'll have a look at that next time. Okay.
Thank you, Stuart. Yeah.
Thank you.
Yeah.
Thanks very much.
Thank you very much, Stuart.
Right. So we can cope with questions if anybody wants to ask any.
Yes, please.
Right. Yeah.
Can you create your own environmental layers?
Creating the environmental layers is something which it would probably be quite useful to cover in another topic. I mean, there's quite a lot of stuff out there that's available now. I think I covered briefly in the first talk some of the sources I've got material from, but there are things, there's governmental sources like the Ordnance Survey. I got the digital elevation model, the altitude data from that. There are things like ITE, which produce the land cover maps. There's the Met Office producing weather data, soil survey, people like NASA and the European Space Agency producing various vegetation maps of land cover and vegetation parameters. So there's a whole range of things. There's a world climate site from which you can get whole world climatic data. That tends to be on a grid of something like 0.3 of a degree, which is 0.1 of a degree is around about 11 kilometers. So 0.1 of a degree is about 10 kilometers square data, roughly. One of the problems, and of course there's the European Union and various statistical data from the European Union. One of the issues about that is you'll find all of these different data sources use different spatial reference systems. So very commonly world data will be on a latitude longitude system, typically using WGS 84, which is the latitude longitude system that the GPS uses. European data is usually on a system called Lambert equal azimuth data, which is a projection that works right across Northwest Europe. OSGB data will be in the Ordnance Survey National Grid. If you go for Irish data, it'll be in the Irish national grid, and so on and so forth. So you end up having to wangle data between different spatial reference systems and having to reproject it. That can get quite complicated. You have to be pretty careful and have a reasonably good knowledge of what you're doing in order not to screw that up when you reproject from one of these systems to another. So it would be a sensible topic for another talk, possibly sometime in the future, if people are interested in how to go about getting hold of these environmental layers. But we are now in a time when there is a lot available, mostly available free. Quite a lot of the sites will require you to actually sign up and create an account, and many of them will ask you for things like some indication of what you're going to use it for, and then if you publish anything as a result of it, they'll want notifying about those publications, and also the data source to be cited in producing that publication. So most of these are sort of academic and research organisations, so they want recognition that their stuff has been used. But otherwise, it is free to download and use. The downloads can be phenomenally enormous. For example, if you go to NASA and you download the shuttle synthetic radar mission data, which is a digital elevation map of the whole of the world, it's about 9 gigabytes of data, which will take you a little bit of time. If you start getting into satellite images to get vegetation index data, you can literally be talking about terabytes to get hold of that stuff. So you need some reasonably good hardware. You need a good internet connection and a large hard disk to actually work on some of this stuff. And actually doing the rehashing of the data can take you significant time and effort. So if you're wanting to get into species distribution modeling, it is probably the biggest part of the issue, is actually creating these layers. And one of the terms and conditions of almost all these sources is you're not allowed to pass on their data. So the fact that I've done it, I cannot, if I stick to their terms and conditions, give you those layers. I mean, how tightly they would worry about these things, I don't know. But if you read the terms and conditions, you can't pass these things on. Oh, thank you. Coffee. Excellent.
Thank you for that. One of the things I was thinking of in terms of building up other environmental layers is, suppose you made an environmental layer which was a model of the distribution of another species, might be another invertebrate, another plant, and then use that as a layer for your model of the distribution of the hoverfly.
Yeah, well, I mean, if you are modelling Portvinia maculata, you would probably want to include the distribution of ramsons in there, for example. Way you might do that is actually do a species distribution model of ramsons and actually use that, the output of that, as an input into a Portvinia maculata model. So those sorts of things are eminently possible. And clearly, if you want species distribution data, you can go to things like the NBN and so on to actually get hold of it. So those things are very possible. And with packages like Terra and Raster, it's very easy to do.
Thank you.
Can I ask a question about sensitivity analysis? So like, I think you implied sort of in the sort of jackknifing and the model itself seems to sort of bring out the key variables, but is there another option where you can kind of look at the most sensitive variables in the equation and sort of see where your model's got the most predictive power?
Well, again, the different systems differ, but most of the machine learning models try to actually identify which are the most important models, which are contributing the most. It's not like if you're using things like general linear models or general additive models, where you can actually partition the variance and actually compute that out and get direct measures of contribution in that way. They are much more puristic sort of things. They're providing estimates, but they do attempt to provide estimates, and certainly as part of the output from things like Maxent, you can see what the variable contributions are. You'd have to go into the Maxent literature to actually see how they go about it. I mean, I've read through it, but I can't say I understand it that tightly. Same's true with things like random forest and boosted regression trees. Again, it's they are indirectly derived. They're not directly partitioned out in the way that regression models are.
So thanks again for the talk, Stuart. I wanted to ask about the ensemble approach that you presented at the end of the talk.
Yep.
So you used 5 different techniques to average across. In your experience, like the techniques that you put into that ensemble, do any of them do better in certain parts of the distribution, or like certain like sparsities of data? Are any of them better at any regions?
There's been quite a lot of literature comparing different modeling systems, and I would say in general, random forest and boosted regression trees tend to come out at the top. They tend to produce a test AUC that is a few percentage points better than Maxent. Maxent tends to do slightly better than things like neural networks, support vector machines, and so on. Things like general linear models and GAMs come out in the next tier. And then you get things like the environmental niche factor analysis and bioclimatic variables that, you know, are a good tier down. They probably 10 or 15% less good in terms of the test AUC. So there's a sort of hierarchy in there. You do find in particular situations and particular species that ranking may change a little. Sometimes Maxent does best, sometimes a random forest does best, but certainly the random forest boosted regression trees and Maxent tend to be pretty close to the top of the tree in stuff that's around at the moment as far as the current literature goes. So an approach to ensemble modeling would be to run a series of each of those, say 3, using different starting positions, because like a lot of this stuff, these can be quite sensitive to the starting data. So just which sample of your presence variables, sample of the background you use will make a difference. And then ensemble, you know, across a group of those. And my limited experience is the ensemble model nearly always outperforms the individual models, but not usually spectacularly so. Maybe 2 or 3, you know, % on the test AUC at the end of it.
It's really interesting to see it explained so clearly. So thank you very much for that.
Right.
Do you have any opinion on, you say there's a bias towards certain areas. I mean, again, I don't know hoverflies that well, but do you have any species where there is a weighting? So, for example, you do have species which have a true disposition towards a certain geographic region, and how have you got any ways of separating out the bias from the weighting?
Well, as far as we know, I mean, there are distinctly northern, southern, western, and so on. I mean, Chrysotoxinum arcuatum that I was using as an example for much of these two talks is basically a northern and western species that doesn't really occur southeast of a line from the Wash up to about the Humber. Whereas there's a second very closely related species, Chrysotoxinum cautum, that occurs entirely on the south and east side of that line. And there are very few squares where both of those species occur, which is very interesting. Maybe competitive exclusion, you never know. There are certainly plenty of species which have got a distinctly southern distribution. There are a number of species that are very distinctly northern Scottish in their distribution. Quite a lot of species that are Atlantic, sort of western. And if you look at their wider European distribution, you'll see them going up Norway and the sort of coast of northern Europe. So yeah, they're definitely from the known points of presence, species that have distinct geographic ranges, and that's why you might want to use this idea of a calibration area when you're trying to model them. In that, if you've got pretty good evidence that there's a fair chunk of your geography that is not occupied, actually using background samples from that area is really not appropriate, and so you would want to try and avoid doing that.
Okay, well that looks as though we've covered everything. Stuart, that was excellent.
You're very quiet, Roger.
Am I? Ah,
It would help if I.
I think I'm getting old. That's really the
Okay,
problem.
you've come back again.
Yeah. Okay. Right. Well, thank you very much indeed for that, Stuart. The critical one, I think, is really your next presentation, which I hope we're going to get a few people in who will be able to interact, because that's really the question. Do the models work? What can we derive from them? And can we really believe them, I guess?
Okay, well, I will stop recording and see you again in 2 weeks' time then.
2 weeks' time, yeah. Good.
Yeah, thanks so much. That was excellent.
Thanks for that, Stuart.
Thank you.
See you soon. Cheers, guys.